Dear students, today we will talk about the oogenesis. I would like to discuss what is the oogenesis. It is the process in which the primary egg cell or ovum becomes a mature ovum. These cells are known as the primary ova. The primary ova remain dormant until just prior to ovulation when an egg is released from the ovary. Here you can see there are two terms. One is the other is genesis. The process of diploid oogonia division and differentiation into a haploid oocyte within the ovary is called oogenesis. On the other hand, I would like to mention here what is the ovulation. You see, when the oocyte is released from the ovary or the mature follicle, rather, it is called ovulation. What may be PGC, the primordial germ cells? The cells which are destined to develop into gametes are called primordial germ cells. The gametes and the primordial germ cells constitute the germ plasm. Germ plasm is of paramount importance because the somatoplasm ceases to exist with the death of the animals. But the germ plasm continues to live on indefinitely in succeeding generations. How these cells are originated? The two views have been given regarding the origination of the primordial germ cells. One is the germinal epithelial cell origin. Walter stated that the primordial germ cells arise from the germinal epithelium of gonads. Other one is the extraordinary gonadal origin. According to this theory, the germ cells arise outside the gonad at an early period of embryo development and then migrate to the gonads. Transportation of primordial germ cells. As the primordial germ cells originate outside, they move to reach the gonads. They are transported in two ways: by bloodstream in chick embryo and by upward movement in human embryo. Phases of oogenesis. Oogenesis is a complicated process. It can be divided into three phases: phase of multiplication, growth phase, and the maturation phase. When we talk about the multiplication phase, it is also called proliferation phase. The germinal epithelial cells detach themselves from the surface and enter into the cortex. These cells are called primordial germ cells. They divide repeatedly by mitosis, and the resultant cells are called oogonia or egg mother cell. The oogonia again divide repeatedly by mitosis. When the division stops, the cells are named primary oocytes. The nucleus of primary oocyte is diploid. The primary oocyte increases considerably by growth. Here you can see the diagrammatic representation of the oogenesis. The primary oocyte, it grows in slightly. It is called the primary oocyte. Due to meiosis first, it divides into one larger cell, which is secondary oocyte, and one smaller cell, which is first polar body. The larger cell again divides into one larger cell, is the mature egg cells, and one polar body. And the first polar body also divides into two cells, which are called secondary polar bodies. For the growth phase, as the name indicates, it is an important phenomenon in the oogenesis. During growth, the nutrients and the other materials necessary for the development of the embryo are synthesized. As the substances accumulated in the cytoplasm, the oocyte increases considerably in size. The growth phase of oocyte is divided into two stages. You can see pre-viterogenesis and the viterogenesis. Viteron refers to yolk. Genesis refers to formation. Before the formation of yolk, the stage is called a pre-viterogenesis. During pre-viterogenesis, the cytoplasmic and nuclear materials of primary oocyte grow and increase considerably in volume. The yolk and other food materials are not synthesized during this phase. The duration of the period of growth varies from animal to animal. Generally, it's a prolonged process. Again, the diabetic representation, how the primary oocyte, secondary oocyte, first polar body, ovum, one ovum, and three polar bodies. The increase in the size of the oocyte is considerable in case of growth phase. The size is proportional to the amount of food reserved in the phytoplasm. In frogs, the young oocyte is about 50 micron in diameter and the mature egg ranges from 1000 to 2000 micron in diameter. So the oocyte increases 20 to 40 times. The hen's oocyte increases 200 times and in mouse it increases 43 times. What about the nuclear changes? The nuclear sap is produced in a large amount. As a result, the nucleus increases in size. The large nucleus of the oocyte is called germinal vesicle. Sometimes the question may be asked, what do you mean by germ life cycle? Again, the homologous chromosomes, they pair together. In amphibians, the chromosomes of primary oocytes acquire a characteristic shape. Thin loops or threads appear on the sides of the chromosomes. These loops give a brush-like appearance to the chromosomes. Hence, the chromosomes are called lamb-brush chromosomes. The loop of these chromosomes are actively involved in the synthesis of messenger RNA. The ribosomal RNA are produced in remarkable amount. They are produced by the nucleolus. As a result, nucleus increases greatly in size. What are the growth of cytoplasmic substances? You can see the mitochondria increase in number. In some animals like the amphibians and the birds, the mitochondria are aggregated to form mitochondrial clouds. The amount of mitochondrial DNA increases, it exceeds even the ribosomal RNA or ribosomal mitochondria. Changes in ER, what is the endoplasmic reticulum? When we talk about the young oocyte, the endoplasmic reticulum, it is in the form of small vesicles. But when it matures, it is in the form of a membranous structure. They take the form of stacks of double membranes, either in parallel or in spiral arrangement. They have no ribosomes, but they contain numerous pores resembling the pores of the nuclear membranes. Changes in the Golgi bodies. In young oocytes, the Golgi bodies are found around the centrosomes. When the oocyte matures, the Golgi bodies form a spherical mass in the mammals. The important function of the Golgi complex, very important point, it is to manufacture cortical granules. Cortical granules are manufactured by the system of Golgi complex. Accessory cells of oocytes. The growing oocytes are surrounded by a special kind of nutritive cells. The cells are immensely assist the growth of oocytes in various ways. There are two types of nutritive cells, namely follicle cells and the nerve cells. Friends, now we come to the next stage, the vitellogenesis. Vitello refers to yolk, genesis refers to formation. The process of formation and deposition of the yolk in the oocyte is called vitellogenesis. Yolk is the nutritive material of the ovum. It is present in the form of platelets or granules. Again, how yolk is originated? When an oocyte starts developing, it does not contain any kind of nutrients. The nutrients are formed only during growth phase of oogenesis. The two theories have been given. Maybe in situ origin. A very small amount of yolk is synthesized in the oocyte cytoplasm. In case of vertebrates, less than 1% only is synthesized by the oocyte cytoplasm. And the exogenous origin, a major part of the yolk is synthesized outside the oocyte. In the vertebrates, it is synthesized in the liver. In insects, it is synthesized in the fat body. In the liver, it is in a soluble state. It is transported by the blood stream to the follicle cells present around the oocyte. The follicle cells deposit the yolk in the oocyte. The transport of yolk to the oocyte is facilitated by the development of finger-like structures. They are called microvilli by the oocyte and microvilli by the follicle cells. Now we come to the last phase, which is the maturation phase. The primary oocyte contains a diploid number of chromosomes, we all know. What happens during maturation phase? This diploid number is reduced to a haploid number by meiosis or reduction division, and the primary oocyte changed into the ovum or egg. This is called maturation. What happens? It can be explained in two types, meiosis first and meiosis second. The mutant divisions are unequal in oogenesis. As a result of meiosis first, the primary oocyte divides into very small, as I had mentioned in the diagrammatic presentation, and a large cell, each with the haploid number of chromosomes. The smaller cell is always formed at the animal pole and is called the first polar body or podocyte. It contains only a negligible amount of cytoplasm. The other cell contains the main bulk of the primary oocyte and is called the secondary oocyte. Dear students, now the meiosis second of oogenesis, it is equal to mitosis. In the second meiotic division, the second oocyte divides unequally into a small and a large cell. The small cell is called the second polar body or podocyte. The larger cell is the ovum. As the second oocyte is dividing, the first polar body divides into two polar bodies. Thus, in oogenesis, I can I can conclude, only one egg is produced along with the three polar bodies. Later on, these polar bodies disintegrate. Thank you very much for patience hearing.